Hey Beaks, it's Mr. Boylan, and today we're going to dive into microevolution and study population genetics. Okay, so let's begin. Here you can see some white-tailed beasties, just like we have here in Pennsylvania. So the evolution of populations, right? One misconception is that organisms evolve. We know that that does not happen, right? Only populations evolve, not individual organisms. Okay, so consider, for example, this finch. During a drought, large-beaked birds were able to survive better, right? Because they could crack open seeds. They had more food available to them. So by means of natural selection, a bird couldn't change its beak, right? But over time, the population shifted and the beak length of birds increased, or the beak size. Okay, so again, birds can't change their beaks, but the population does change. So microevolution is a change in allele frequencies in a population over generations. And three mechanisms that cause allelic frequency to change, well, natural selection, of course, right? And then we're going to talk about these two later, genetic drift and gene flow. Basically, this is just chance of something happening to change a allelic frequency. And then gene flow is basically like immigration or emigration. Okay, but I'll have other videos on those eventually. But only natural selection causes adaptive evolution, meaning that natural selection is the thing that's going to cause organisms to become better fit for their environment. Genetic variation makes evolution possible. We know that there has to be variation in a population for evolution to happen now, right? And all you had to do is look back at Mendel's work with pea plants and see that, oh yeah, that's genes, right? And genes can be different from organism to organism inside of a population. Okay, so you know how we vary from each other, and that's because of our sequence of DNA, right? Our nucleotides, and the phenotype is the product of this inherited genotype, and we have environmental influences as well. Okay, but natural selection will only act on our genetic components. So you know that we have both discrete and quantitative characteristics. So discrete characteristics are an either or. That's kind of like a pea plant having purple or white flowers, right? So an either or kind of thing. Either you have a widow's peak or you do not. Whereas quantitative characteristics vary along a continuum, if you remember, like polygenic inheritance, right? So sometimes it's the amount of certain alleles that you get in certain genes that will give you your traits. So genetic variation can be measured as gene variability or nucleotide variability. Okay, so for gene variability, you can, and we're going to look at both of these today. You can, for example, look at something like average heterozygosity, okay, in a population. So how many people in a population are heterozygotes? Sure, we could look at something like that. Or we can actually sequence people's DNA and actually look at the ATCs and Gs and see what's going on there as well. So we could look at nucleotide variability. Okay, so there is variation between populations, of course. I'll just tell you, humans are a little tougher with this, right? Because we travel all over the planet and we mix it up nicely. So it's a little tougher to see with humans anymore, but most species exhibit some sort of geographic variation, as do humans, of course. For example, you have this little island off of Portugal, I believe it's Madeira, and it has these mice on there. And as you can see, their chromosomal variation is due to drift, not natural selection. And what that means is that these mice populations here, okay, same species, but they are different from each other. If you notice here, their chromosomes are very different, but it's not because nature came in and acted differently. This is pretty much all the same environment here. Okay, so like I said, we'll get into genetic drift later, but it's not like nature selected something good to happen or something different to happen because of these chromosomes. It's just random chance. So depending on where you live, populations can change, right, differently from other populations, even of the same species, if you're not connected to each other. Okay. Some examples of geographic variation occur as a climb, okay, a graded change in a trait along a geographic axis. So this mummy chog minnow here, you can see that if there's a allele along a temperature gradient, right? So it's a cold adaptive allele. So as you can see, the allele frequency goes up if you live in a colder climate, okay, or colder area. Whereas if you live over here where it's warm, well, you don't have that as high of a frequency. Okay, makes sense. So where do we get this genetic variation from? Well, the main thing, of course, is mutation. You could also have gene duplication, but mutations are those random changes that happen, and they could be caused because of mutagens and things like that, but a lot of times it's just your DNA changing. So Yes, mutations are a big deal, and as you also know, genetic variation for sexually reproducing species, oh yeah, this is hugely important, right? Remember meiosis in prophase 1 we had crossing over, and in metaphase 1 we had independent assortment, and then random fertilization. So let's quickly review that.
In sexually reproducing organisms, three processes lead to most genetic variation: independent orientation of chromosomes in meiosis, crossing over of chromosomes in meiosis, and random fertilization. Each pair of homologous chromosomes consists of one chromosome inherited from the father and one from the mother. Here we have color-coded them blue and red. Each pair of chromosomes lines up independently of the other pairs in metaphase 1 of meiosis. Here you see one of the possible arrangements and outcomes. There are two different ways that each chromosome pair can line up. That means that in the organism shown here, with the diploid number of 4, independent orientation of chromosomes at metaphase 1 can produce gametes with 4 different combinations of maternal and paternal chromosomes. In a human being with 46 chromosomes, more than 8 million combinations are possible. Now let's look at how crossing over creates even more genetic variability. During prophase 1 of meiosis, homologous chromosomes pair up very closely, and corresponding parts of two non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from two parents. Here the process produced a total of 4 genetically different gametes. There are many ways crossing over can occur. In humans, crossover events happen an average of 2 or 3 times per chromosome pair, greatly increasing the variation among eggs and sperm. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes. Independent orientation and crossing over occur simultaneously during meiosis, multiplying the number of genetic variations among gametes. Because each pair of chromosomes lines up independently, and crossovers can occur almost anywhere along each pair of chromosomes, it is possible for a human being to produce an almost infinite variety of gametes. A sperm fertilizes an egg, producing a zygote. The random nature of fertilization adds to the variation arising from meiosis. Each parent is capable of producing a huge variety of genetically different gametes. The number of possible combinations among their offspring is staggering. Theoretically, one human couple is capable of conceiving a number of genetically different offspring that is far greater than the number of humans who have ever lived.
That's crazy, right? Where you're watching me, watching it. Amazing. So yeah, there's lots of variation in population because of sure mutations in our gametes, right? But also those things that we do in meiosis, right? Where we create gametes and the random fertilization that has to happen, that's amazing. So we're very lucky to have so much variation in our population. Okay, so formation of new alleles. A mutation is a change in a nucleotide sequence, right? Remember we learned all about point mutations, which can be, silent, or you can have, excuse me, I'll put myself up here. You can have point mutations that are silent, where you didn't change the amino acid. You could have missense, where you change the amino acid, or you could have nonsense, where you hit an early stop codon, or you could do a frame shift mutation, and that changes the whole reading frame for gene expression, right, for translation. So formation of new alleles does happen, and it tinkers, right? Microevolution is a way to tinker with nature's toolbox, and that toolbox is basically our genome, right? So mutation is this change in a DNA sequence, and again, only when we change our gametes will that be passed on. So if I have mutations in my somatic cells, my offspring shouldn't inherit those mutations. And like we said here, you can see in sickle cell, well, if the template strand of DNA changes by one letter, and now my mRNA, of course, will change. Well, glutamic acid gets changed to valine, and that can have severe, repercussions with sickle cell anemia. Okay, and like we said, it matters, and you only use about 2% of your DNA for these protein coding genes, but again, it can be a big deal. Now, they can be neutral, like we said, because remember the redundancy of the code, where you have 64 possible codons. but you only have 20 amino acids being coded for, and that's because of that wobble pair, if you remember that, the third base. So with the tRNA and the mRNA. Anyway, all right, let's continue. The effects of point mutations can vary. Again, they could be harmful, but every now and then, maybe you'll hit something that's beneficial, right? There's a reason that we're very good for our environment right now, and it's because of mutations. And you would think that we're set up right now for our environment, so unless there's a huge change, that a chromosome or a DNA mutation would be a bad thing, but not necessarily. Maybe some of these changes that are happening are actually beneficial change, making better shaped proteins or whatever their job is. Of course, you can also alter the number or position of genes on chromosomes. So we learned about these before. Remember, chromosomal mutations, they're an even bigger deal, right? Because they can goof up huge pieces of DNA. So an example here, the odor detecting gene has been duplicated many times. So in this case, this is a good thing. Gene duplication, right? Humans have about 1,000 copies of this gene, whereas mice have 1,300. Okay, so that's an example there where we're getting better, or we can smell things, detect odors better, right? All because we have a lot more copies of that gene. Mutation rates are somewhat low in plants and animals. It's an awkward statement because they still happen quite a bit. The average is about 1 mutation per 100,000 genes per generation. Okay, yeah, but don't forget, you have, 6 billion bases, 3 billion base pairs, so yeah, mistakes will happen. They will happen. It just so happens that hopefully they don't happen at a very important spot. Okay, and mutation rates are lower in prokaryotes and higher in viruses, and that's something that we're dealing with right now, right? We're home because of the coronavirus, and we're hoping, as we're praying for a vaccine to come soon, that this germ, this pathogen doesn't mutate very much. So sexual reproduction, then, again, can shuffle existing alleles into new combinations. What we just saw there, this how you line them up and how you shuffle them together. And you're basically taking your mom and dad's DNA and randomly shuffling it when you go to reproduce, right? So when you have a child someday, what are you doing? You're randomly taking the two people that made you and just kind of dealing out a random shuffle of those genes, because you're only allowed to give one copy, right? That's a haploid gamete. And this is hugely important. It's actually more important than mutations, right? The fact that we reproduce sexually means that our kids are going to be a nice mishmash of variation, right? We don't want to just rely on mutation because that would be problematic. Okay, so here we go. We're going to talk about the Hardy Weinberg Equation and how it can be used to test if a population is evolving or not. All right, so basically here you can see we have Godfrey Hardy. Okay, if I can, just a great and powerful name and a great picture sitting there. He was a mathematician from Cambridge. And here we have an obstetrician, right? A general practitioner obstetrician. Push! This is Wilhelm Weinberg. Okay, so Hardy and Weinberg, what a pair, came together and created this theorem that we're going to use to see if populations are evolving. Okay, or to say when populations won't be evolving, basically. And to start understanding the Hardy-Weinberg equation, we have to know exactly what a population is. All right, so gene pools and allele frequencies. Population is a localized group of individuals capable of interbreeding and producing fertile offspring. Sure, so whatever of the same species, whoever lives in a certain area and can reproduce with each other and produce offspring that can also reproduce, sure. Okay, you're a population. The gene pool then is all of the alleles in that population. Okay, so alleles can become fixed if they're homozygous. So for example, let's take a look at these, what are these anyway, warthogs, I guess? Let's take a look at these things and say, let's pretend that they're all little b, little b. Well, if these are all little b, they're all homozygous recessive, it doesn't matter what happens here for that gene, right? Because all the alleles are recessive, that all the kids born are going to be little b, and little b will be all that ever happens here until something perturbs that system. Same thing if they're all big B, homozygous dominant, right? So the fact that we have variation, okay, in our genes and these different forms of a gene or alleles, okay, makes that variation more apparent in our populations. But it can become fixed, okay? So the frequency of an allele in a population can be calculated. For diploid organisms, the number of alleles at a locus is the total number of individuals times 2. That makes sense, right? So if you think about it, whatever gene you want to talk about, let's talk about like earlobe attachment or whatever, everybody has 2 copies. Sure, we're diploid. We got one from our mom, one from our dad. So the diploid organism, the total number of all alleles is the number of people, in this case, times 2. Okay, the number of dominant alleles at a locus is 2 alleles for each homozygous dominant. That makes sense. And then add 1 more allele if you're a heterozygote, right? Because let's go back to earlobe attachment. Again, all of the people who have attached earlobes, okay, that would be all little e. But don't forget to go ahead and add in the heterozygotes also have 1 little e, right? So it would be the opposite, let's say free earlobes. If we knew who had big E, sure, but then you also have to add in the 1e, big E from all the heterozygotes. So you just have to make sure all the alleles are accounted for, okay? So by convention, if there are 2 alleles at a locus, P and Q are used to represent their frequency. And the frequency of all alleles in a population have to add up to 1. Well, that makes sense, right? So if you add up every single allele, okay, that would be 100% of the alleles in a population. All right, and we use frequencies here. It's like basically percentages. So let's take a look at a population of wildflowers that is incompletely dominant for color. Okay, so first of all, you have to go back to your genetics. Remember what incomplete dominance is? That's when the heterozygote is a blend, right, of the 2 homozygotes. So the homozygous dominant, the homozygous recessive, the heterozygote is kind of when they're blended together. So in this case, red and white make pink. So if you have 320 red flowers, okay, and notice that's homozygous red, and if you have 160 pink flowers, which means, okay, we have red and white here, alleles, and then 20 white flowers is, of course, just homozygous recessive white. So calculate the number of copies of each allele. Well, this is what we were just talking about. So for the red allele, you have 320 right here. Oh, but it's times 2 because we're diploid. So 320 times 2, and then, oh, don't forget, there's also a heterozygote. So 160 as well because of that guy right there. Okay, so that equals 800 copies of the red allele in this population. And the white is, of course, 20 times 2 because we're diploid. And then also 160, so we have 1 there. Okay, so that's 200. So in this population, we actually have 1,000 alleles. So to calculate the frequency of each allele, pretty simple stuff here, right? How many alleles out of the total are there? So in red, there were 800 out of 1,000, and that equals 0.8, right? Or 80% of the alleles would be red, okay? And that's your P, okay? The frequency of the dominant allele is your P, whereas your Q is the frequency of the recessive allele. And in this case, we have the data, so it's 200 out of 1,000, so about 20%, right? 20% of the alleles are the recessive variant. Okay, so the sum of alleles is always 1. 0.8 plus 0.2 equals 1, right? Or 80% plus 20% equals 100% of the alleles, if you want to think of it that way. So the Hardy Weinberg principle describes a population that is not evolving. Okay, so Hardy and Weinberg came up with this idea of how would evolution not happen. Okay, and if a population does not meet the criteria of the Hardy Weinberg principle, it can be concluded that the population is evolving. So obviously, we are evolving, right? But what we're trying to show here, what Hardy Weinberg is trying to show is that, well, if you're not evolving, here's what has to happen. Let's take a look. So it states that frequencies of alleles and genotypes in a population remain constant from generation to generation. In a given population where gametes contribute to the next generation randomly, allele frequencies will then not change. Okay, which is, again, very, very unlikely. What we're talking about here is craziness. But let's see, Mendelian inheritance preserves genetic variation in a population. Well, of course, sure. We know the rules of Mendel. So here we go. Hardy-Weinberg equilibrium describes the constant frequencies of alleles in such a gene pool. Consider, for example, the same population. So we just figured out our P's and Q's for these wildflowers, and we figured out that the frequency for the dominant allele was about 0.8. Okay, and well, not about it, exactly 0.8, and the frequency of the recessive allele was 0.2. Okay, whoops. So I'll give you a preview there. So this is the alleles in a population. Here are all the flowers, right? And what are those flowers going to do? Well, eventually they're going to reproduce. And what exactly are your chances of reproducing different things here? Well, think about it. If 80% of the population has, or of the alleles of the population are red and 20% are white, we'll look at the egg then. It's going to be about an 80% chance of putting red in there and a 20% chance of putting the white allele in there. Same thing for sperm. So now when we move ahead here, okay, the frequency of these things can be calculated. All right, now this is a little bit odd, right? So think about this. The frequency of genotypes. Now we have not discussed this before. All we've talked about up to this point has been the frequency of the alleles in a population, right? We saw how many red alleles and white alleles there were. But now what we're doing is we're going to look and see how many actual genotypes there are. So we're talking homozygous dominant, homozygous recessive, and heterozygotes in a population. Okay, and that's the beauty of this Hardy Weinberg. When you get into it, you can calculate all of this stuff, assuming that this population isn't changing. So here we go. How many of these flowers in this population, okay, are homozygous dominant, right? Okay, well, let's see here. We have 0.8, and to be homozygous dominant, that would be your big R, right? 0.8 times, 0.8, okay, that's your p squared. So theoretically, 64% of the population is homozygous dominant. And now what about your pink? Okay, well, this is going to be 2pq, right? Because what it takes here is for one parent to give the red and one parent to give the white. Oh yeah, but also the other parent could give the red and the other parent could give the white. So it happens in two different ways here. Okay, and that's going to be 32%. And finally, what are your odds of having a white? Well, white was 0.2, and q squared is going to be our recessive, homozygous recessive individuals in a population. So that's about 4%, or 0.04 is the frequency of that genotype, right? And we can confirm this using a Punnett square. As you can see, some people really love their Punnett squares. Okay, so take a look. Right, so theoretically, in our population, if it's about 80% red and red, well, yeah, about 64%, and you can take a look at this, and that should make sense to you. Okay, so if P and Q represent the relative frequencies of the only two possible alleles in a population in a particular locus, right? So we're not talking about multiple alleles here or anything crazy like that. We're just talking about the only two possible alleles at that gene locus. Then p squared plus 2pq plus q squared equals 1, right? And again, this is where p squared is going to represent your homozygous dominant, and q squared represent the homozygous recessive. 2pq, okay, this is your heterozygote in a population. And if you added up for, again, a gene with only two possible alleles at a certain locus, if you added up all your homozygous dominants, all of your heterozygotes, and all of your homozygous recessives, that's going to be 100% of the people here, right? So hopefully that makes sense. So the conditions for this to happen, okay, is a hypothetical population that's not evolving. Okay, so obviously, you know, this is hypothetical. Normal populations will, of course, be evolving, okay, even minutely. But this is saying that, okay, let's see what has to happen here for populations not to evolve, okay, even though we know that they do. So there are 5 conditions, okay, that have to be met for non-evolving populations, and these things would almost never be met in nature. Okay, no mutations, random mating, no natural selection, extremely large population size, and no gene flow. Okay, so natural populations can evolve at some loci while being in Hardy Weinberg equilibrium at another loci. And I mean, all that means is this: what if we had an island of people, and on this island, every single person that lived there had attached earlobes? They were all little e. Well, sure. So that's now fixed. So on that island, through the generations, as long as, you know, nothing crazy happens, theoretically, little e is going to be what it is. But that doesn't mean that there's not other genes or alleles that are changing, right? So evolution is still happening, maybe not at that one locus, but at other loci, sure, we're changing still. So here are those things that have to be met, those Hardy Weinberg, like we said here, the conditions that have to be met. Number 1 is no mutations, right? If a gene mutates from one form into another, you can create these new alleles, can be put into the population, right, in the next generation, and the allelic frequencies will change then. So obviously we don't have a way to turn these off, okay? But if you wanted to have no evolution in a population, we would have to have no mutations. So already, right out of the gate, we can see that this is crazy, right? We're not really going to be able to hit this in nature, but this is theoretical. Okay, so no mutations. Okay, number one, good. Number two, random mating. Now, hopefully, you all remember that this is also crazy. You know that in nature, and again, don't think so much humans, but think of other populations in nature. No, mating is often not random. A lot of times mating is very choosy, right? And sometimes only certain individuals in that population get a chance to mate. A lot of times it's based on their characteristics, right? So for Hardy Weinberg's theorem to be true, all mating has to be random. Okay, it ensures that each individual has an equal chance of passing on its alleles to its offspring. You know that that's not necessarily very true. Okay, number three, no natural selection. Okay, so all genotypes in a population have equal probabilities of surviving and reproducing. You know that this is not true either, right? Just look at those tigers right there, right? If this was in nature, you'd know that, well, one of those tigers is probably very camouflaged and will get more prey and things like that, and the other one probably won't. Okay, so no phenotype can have a selective advantage here. No natural selection can happen. Okay, this one is very, very important. You have to have an extremely large population. Okay, now we haven't really gotten into genetic drift yet, but basically all this says is chance plays a role in evolution if the population is very tiny. But if you have a gigantic population, then chance is kind of mitigated to an extent, okay? So basically, for Hardy Weinberg's theorem to be correct and evolution is shut down, is not happening, you would need a very, very large population, okay, to keep that equilibrium going. Okay, and finally, no gene flow. Okay, and we'll talk about this a little later too. But basically, this one should make sense, right? If you're talking about no allelic frequencies changing, no evolution happening in this population, well, then you can't have other people immigrating into your population, and you can't have some of your own population emigrate out either. Theoretically, let's keep it the way it is right now, and that won't allow the allelic frequencies to change. Okay, so that's what Hardy Weinberg finished up with, saying, yeah, if you meet all five of these, that theoretically should mean no evolution is happening. And again, not very likely in nature. So let's try and solve something here. Let's do a problem together. Applying the Hardy Weinberg principle. Okay, we can assume that the locus that causes phenylketonuria is in Hardy Weinberg equilibrium. So remember, we're doing genetics. This is an autosomal recessive genetic disorder. Okay, and remember, these kids can't have too much phenylalanine in their diet, right? Because it can lead to, it's pleiotropic, it can lead to a whole bunch of problems if they have too much of that. PKU gene mutation rate is very low. Okay, so we're going to assume that this is in Hardy Weinberg equilibrium. Just this one gene, not necessarily the whole population, but we're looking at one gene at one locus here. Okay, the PKU gene. And we can assume, okay, the PKU gene mutation rate is very low. Okay, so what else? Well, mate selection is random, right? Most people aren't out there running around asking, you know, before they decide to reproduce about PKU. Okay, so theoretically, that's not one of the reasons that you would or would not mate with somebody. All right, so we're good there. And if you have a chance, read up about how this test for PKU came about. It's a really cool story. Okay, and then natural selection can only act on rare homozygous individuals who do not follow dietary restrictions. All right, so natural selection, yeah, it will be active in a population, but think about what this is saying here. It's saying that if you are one of those rare kids that are born, which happens, right? One of those rare kids that are born with little p, and you are a phenylketonuriac, that's okay, because as long as you follow the dietary restrictions, as long as you have access to health care and your parents are watching out and your doctor's telling you what to do, you should be just fine. So natural selection really doesn't mess too much with this population. Okay, the population is large. Well, yeah, we're going to be talking about the United States as a whole here for this problem. So that's a gigantic number of people. Okay, and migration has no effect, as many other populations have similar allelic frequencies. So yes, we're talking about the United States, but whoever comes in and whoever goes out, the entire globe pretty much has similar numbers here. And this is another example of how, why do we still have this in our population? There might be some underlying meaning to it or reason for it, but for the most part, we're diploid. So it's really hard to get rid of an autosomal recessive disease because it just hides in carriers. So theoretically, the whole world has it in the same numbers basically that we do. Okay, so here we go. We always will get in our problems, whenever you're solving these Hardy Weinberg problems, there's always going to be something given to you to start. Okay, and in this case, let's see what's given to us. Okay, the occurrence of PKU is 1 per 10,000 births. All right, so what did they just give us there? 1 out of 10,000 kids will have PKU in a population. Oh, what that is homozygous recessive, right? So they gave us Q squared. Okay, that was given to us, and now we're ready to roll with this problem. Because we heard that 1 out of 10,000 is little p, that means we got our Q squared value right here. And how did we get that value? Of course, 1 out of 10,000, okay, or 1 divided by 10,000. Here you go, 0.0001 is our Q squared. That means in a population, that many, that's the frequency of homozygous recessive, or in this case, sufferers of PKU. Now we can get our Q from that, right? Square root, sure, that's easy enough, 0.01. And now hopefully you know what that number means. This means that the frequency of not people here, not organisms, but the allele. This is the allele frequency for little p. The recessive allele is 0.01, or about 1% in the population. Okay, so now the frequency of normal allele, well, this should be easy. We know that p plus q equals 1, right? So that means that we have q. So that simply means that, oh, okay, if p plus q equals 1, then p equals 1 minus q. So 1 minus 0.01 equals 99%. Okay, so that's our p. That means p is 0.99, or 99% of the allele in the population is big P. So now the frequency of carriers, oh, easy stuff, 2pq, right? Because it could be from either parent. So 2 times p, which is 0.99, times q, that means our 2pq. So now we're not talking about allelic frequencies here. Okay, those are p's and q's. We're talking about 2pq. We're talking about, in the United States, how many people, okay, in this population carry or are heterozygous, okay, for this disease, and it would be about 2%, right? So there's an example where if you had a test question or on the AP exam or whatever you're taking, SATs, anything, and it says, hey, what % of the population of the United States carries the allele for phenylketonuria, you'd say, well, where do I start here? There has to be something that's given to me so I know where to start, and in this case, they gave us Q squared. And as you see, once you're given Q squared, you just work backwards. Okay? So that was a little bit on population genetics and intro to Hardy Weinberg, and we'll try some problems together, and let me know if you can clarify anything from this video, and I hope you found it helpful. All right, take care.